So, um, if you think about a typical medical diagnosis, there's usually this expectation of, I don't know, pure mechanical precision.
Right, yeah. Like, it's a straightforward binary.
Exactly. You fall, your arm hurts, and you get an X-ray. The doctor pulls up that image, points to a jagged white line on the screen, just says, there it is. That's the problem.
And we find a lot of comfort in that kind of visibility, you know? We want biological problems to be broken or not broken,
Yeah.
sick or healthy.
But then you step into the world of oncology and targeted drug delivery. And that comforting precision just completely vanishes. The landscape becomes incredibly murky.
Oh, absolutely.
Because one of the most frustrating challenges in modern medicine is this. What happens if you have a perfectly designed nanomedicine, like a brilliant targeted therapy, but you simply cannot get it to physically enter a solid tumor?
Yeah, it represents, well, the ultimate biological bottleneck. I mean, you can spend a decade designing the smartest, most lethal, cancer-killing drug in the world. But if it just bounces off the outer armor of the target tissue, all that effort is essentially useless.
So welcome to this deep dive. Today we are taking a stack of sources you sent us, which, by the way, is a really fascinating mix of dense cell biology research, oncology papers, and some cutting-edge nanomedicine studies.
Yeah, some really heavy reading in there.
Seriously. But we are unpacking them for you today, and they all orbit this single massive medical problem, which is infiltration. Basically, how do we get the medicine in?
So our mission today is to explore a very specific, almost deceptively low-tech intervention.
Yeah, this part blew my mind.
We're going to look at how applying a precise, mild amount of local heat completely rewrites the physical and biological rules of human tissue. We will break down how preconditioning a target area with this heat drastically alters two massive roadblocks.
Which are local blood flow and the cellular uptake of these biological delivery vehicles called exosomes. I want to start by understanding the baseline, though. Because before we get into how heat breaks the rules of the body, we really need to understand what those rules actually are.
That's a good place to start.
And reading through these papers, particularly the ones focused on the blood vessel linings, I just kept visualizing a fortress wall.
The baseline state of your blood vessels is perfect for that analogy because they're definitely not just passive, leaky plumbing pipes.
Right.
Your vessels are lined with endothelial cells. Their entire function is to form a highly strict, highly regulated barrier to keep the blood inside. They prevent random molecules from just washing out into the surrounding tissue.
So if we imagine those endothelial cells as the literal bricks in this fortress wall, they can't just be randomly stacked on top of each other.
No, not at all.
The research kept pointing to this specific signaling complex centered around something called VE cadherin.
Vascular endothelial cadherin, yeah. Think of VE cadherin as the primary cellular glue. Or, keeping with your analogy, the molecular mortar that spans the space between adjacent endothelial cells.
It physically links them together.
Right, it links them, but the crucial concept to grasp here is that this mortar isn't like concrete that dries and sets permanently. It requires constant active management by the cell to maintain its integrity.
Well, let's build on that masonry analogy because I found the mechanism here so interesting. If the endothelial cells are the bricks and the VE cadherin is the mortar, there's another protein mentioned in the research called trio.
Ah, yes. Trio.
And the way trio functions, it feels like the master mason of this whole operation.
The master mason is a really helpful way to visualize it. What the research shows is that trio binds directly to the VE cadherin mortar, and then it does something highly specific. It locally activates another protein called RAC1.
And RAC1 acts like the scaffolding crew working under the master mason.
That captures the dynamic perfectly, actually. When trio activates RAC1, that scaffolding crew reaches deep inside the cell and physically rearranges the cell's internal skeleton.
Which is the actin cytoskeleton, right?
Yeah. Exactly, the actin cytoskeleton. It takes these messy radial actin fibers and binds them into strong, highly organized bundles that run parallel to the cell junctions. It essentially reinforces the wall from the inside out, locking that VE cadherin mortar down tight.
So the whole system is active. The mason is always working, the crew is always building. But, what happens if something disrupts that? I know the researchers did experiments where they silenced or removed trio to see what would happen.
Just results were immediate and honestly chaotic. When they removed trio, the master mason, the wall fundamentally destabilizes.
Wow.
Those smooth, continuous, tightly locked junctions between the cells just degrade. They turn into what the literature calls focal adherens junctions, or FAJs.
Meaning the bricks basically start shifting around and separating.
Yeah, the connections become incredibly irregular. They start frantically assembling and disassembling in this unstable loop. As a result, the vessel becomes highly permeable, or leaky, and the electrical resistance of that cellular layer completely plummets.
Okay, so this is a vital baseline for you to keep in mind as we go. Your blood vessel walls are not a static barrier. They are a highly regulated, active structure constantly responding to their environment.
Which brings us to the core problem of drug delivery.
Right, because if this trio and VE coherence system is actively locking down the fortress to keep things inside the bloodstream, how do we intentionally breach it?
Yes.
If we want our cancer drug to exit the bloodstream and leak out into the tumor, we have to find a way to essentially dismantle the wall.
And this is where intervention comes in. The thermal breach.
Here we go.
The first major effect of our targeted heat is to intentionally alter that local blood flow and override the barrier permeability.
But I want to emphasize, the sources make a massive distinction about the type of heat. We are not talking about cooking the tissue.
No, definitely not.
The sweet spot here is termed mild hyperthermia, which is strictly defined as heating the tissue to a range of 41 to 43 degrees Celsius. So for those of you listening, that is roughly 105 to 109 degrees Fahrenheit.
Yeah, we are deliberately avoiding thermal ablation. Ablation is where doctors use extreme temperatures, like above 45 degrees Celsius, to literally burn and cause direct necrosis or death to the tumor tissue.
Right, that's just destroying it.
Exactly. Mild hyperthermia operates on a completely different, much more subtle biological level.
Now, if you're listening to this and wondering why on earth we'd want to heat up cancer, you are not alone. I mean, think about the last time you used a heating pad on a sore muscle.
Right, it causes vasodilation.
Exactly. Heat opens up the blood vessels and increases blood flow. So my immediate thought was, wait, wouldn't heating a tumor to 42 degrees just flood it with fresh blood and oxygen? Aren't we just giving the cancer exactly what it wants to grow? I really struggled with this paradox when I was reading.
I mean, your logic is perfectly sound, and it highlights one of the most fascinating paradoxes in oncology. Solid tumors are notorious for growing so aggressively that they outpace their own infrastructure.
They don't build neat plumbing.
No, not at all. Their blood vessels are a chaotic, tangled mess of dead ends and inefficient loops. And because of this terrible plumbing, large sections of the tumor become deeply hypoxic, meaning they are utterly starved of oxygen.
And from what I understand, an oxygen-starved tumor is an incredibly stubborn tumor.
Hypoxia makes cancer cells aggressively resistant to traditional treatments. Radiation therapy, for instance, literally requires oxygen in the tissue to create the free radicals that ultimately shred the cancer's DNA.
Oh, wow. So no oxygen means the radiation just passes through without doing its job.
Pretty much. But by applying mild hyperthermia, we force those tangled, inefficient vessels to dilate. Oxygen rushes into those dead zones, and suddenly the tumor's resistance to radiation drops drastically.
But we aren't just trying to deliver oxygen here. We are trying to deliver a physical medicine.
And that increased blood volume acts as a hydraulic flushing mechanism, driving chemotherapeutic agents deep into the tumor bed. But more importantly for our nanomedicines, the heat is physically acting on that trio and VE cadherin fortress wall we talked about earlier.
Okay, so the heat starts interfering with the master mason.
It creates profound disruption. When tissue reaches that 41-degree threshold for just 1 hour, the thermal energy induces a physical breakdown in the highly regulated actin cytoskeleton.
So the scaffolding crew falls apart.
Right. The signaling gets confused, the crew loses coordination, and literal physical gaps are pried open in the endothelial lining.
I was absolutely floored by the scale of this. The sources note that these thermally induced gaps can stretch up to 10 micrometers wide.
It's massive on a cellular level.
Yeah, to give you a sense of scale, a standard red blood cell is only about 8 micrometers across. So this mild heat is opening doors in the vessel wall large enough to drive entire cells through, which means our microscopic nanomedicines can easily flood through those gaps.
And the clinical utility of this is even more impressive because the effect isn't momentary. Those massive gaps do not simply snap shut the second you turn off the heat source.
Right.
The data indicates that this window of high permeability remains open for up to 8 hours after the mild hyperthermia treatment concludes.
An 8-hour window where the fortress gates are just locked open.
Yeah.
That completely changes the timetable for drug delivery. There's a specific animal study in the sources that really drove this home for me using ultrasound imaging.
Oh, you're referring to the in vivo study on mice with breast
Oh,
tumors.
yes, that one.
So the researchers applied mild microwave irradiation to the tumor area to generate the heat, and then they utilized contrast-enhanced ultrasound to physically measure the functional blood vessel space.
And what did they find?
The imaging proved that the total accessible area of the tumor blood vessels increased by a factor of 4 following the mild heat treatment.
4 times the surface area for the drug to leak out.
That is wild. Okay, let's pause and just map out where we are in this process. We've applied our 41 to 43 degree heat. The trio and V cadherin bricks in the blood vessel wall have been pried apart. The sudden surge in blood flow has flushed our delivery vehicles out of the bloodstream through those massive 10-micrometer gaps, and they are now floating deep inside the interstitial fluid of the tumor tissue itself. So the first major bottleneck has been bypassed. Extravasation is successful.
But they're just floating in the fluid outside the target cancer cells. How do they actually force their way inside the cell? Because to understand this, we need to define what these delivery vehicles are. The research relies heavily on something called exosomes.
Right. Exosomes are fascinating structures. They are tiny, naturally occurring biological bubbles, specifically lipid vesicles.
Almost every cell in your body naturally secretes them, right?
Exactly. You can think of them as microscopic biological cargo ships. Cells use these bubbles to package up proteins, RNA, and other molecular instructions and mail them to neighboring cells.
So we are basically hijacking the body's own internal postal service to deliver the medicine.
It's an elegant strategy because exosomes are native to the body. They are naturally stealthy, meaning the immune system largely ignores them. This makes them an ideal Trojan horse for smuggling cancer drugs.
But even a Trojan horse has to get through the final gate. And as I read the mechanics of this, I realized it's not like watching 2 soap bubbles bump into each other and just effortlessly merge into one bigger bubble. Fusing with a cancer cell actually takes work.
It requires a significant amount of cellular energy to overcome what we call the activation energy barrier. Normally, a cell has to actively consume these exosomes through highly orchestrated processes. One of the primary methods is called clathrin-mediated endocytosis.
Can you break that down? Like, how does clathrin-mediated endocytosis actually work when an exosome arrives at the cell?
Well, when the exosome docks at the cell membrane, the membrane dimples inward. Proteins called clathrin begin to assemble on the inside of the cell membrane, linking together to form a literal geometric cage around that dimple.
Cage.
Yeah, and this clathrin cage physically pulls the membrane deeper into the cell until it pinches off, creating a new bubble inside the cell that contains the exosome. It's a complex, highly energy-demanding piece of cellular machinery.
The sources mentioned a really wild detail about how the exosomes even find the right spot to trigger that cage-building process. Like, they don't just blindly crash into the side of the cell. They actually surf on something called filopodia.
The cellular topography is incredibly dynamic.
So if you were picturing a cancer cell, don't picture a smooth sphere. Picture a cell covered in these microscopic, reaching, active tentacles. Those are the filopodia. And the research states that the exosome lands on one of these tentacles and literally surfs down the shaft to find an endocytic hotspot at the base.
Which is where the clathrin machinery is waiting to swallow it.
Right.
But again, the cancer cell membrane is a tough, rigid lipid bilayer. For that clathrin machinery to pull it inside, or for the exosome to fuse directly, it has to overcome the physical resistance of those packed lipids.
So how does our mild heat treatment help with this specific hurdle? We know it breaks the masonry of the blood vessels, but how does it force the cancer cell to swallow the drug?
Well, this brings us to the second major mechanism of our thermal preconditioning. Heat alters the fundamental physics of the target cell's outer membrane.
Okay, how so?
Think about the basic physics of kinetic energy. When you heat up a lipid like butter in a frying pan, the molecules move faster. They push slightly further apart from one another. By applying 41 to 43 degrees Celsius to the tumor cell, we significantly alter the fluidity and the rigidity of its lipid bilayer.
So we are essentially melting the tumor's armor.
We are radically lowering that high activation energy barrier. The membrane transitions from a rigid, tightly packed state into something much more fluid and dynamic. In this heated state, the cell membrane becomes highly susceptible to endocytosis. The physical resistance to being dimpled and swallowed just drops.
There was an incredible study in the sources that tested this concept using something called LICUS, but I wasn't entirely sure how the specific technology functioned.
LICUS. It stands for Low Intensity Continuous Ultrasound. It's a brilliant application of physics in a clinical setting. While we typically associate ultrasound with diagnostic imaging, like looking at a fetus, therapeutic ultrasound interacts with human tissue in much more aggressive ways.
Right.
In this specific study, they operated the ultrasound at a frequency of 3 megahertz in vitro.
So what does a 3 megahertz sound wave actually do to a cancer cell?
Well, at that frequency, it generates two simultaneous forms of stress. First, it generates our required mild hyperthermia, hitting that 42 to 43 degree sweet spot.
And the second?
Secondly, it creates mechanical stress. The sound waves are physically massaging, pushing, and vibrating the cell membrane.
So the cell is being heated into a fluid state while simultaneously being physically hammered by sound waves.
Exactly, and this triggers a phenomenon called mechanotransduction. The physical force of the ultrasound pushing on the cell membrane is translated by the cell into biological signals. In the study, researchers took exosomes derived from hypoxic liver cancer cells and loaded them with a natural, highly hydrophobic therapeutic drug called curcumin.
Hydrophobic meaning it repels water, right? So tucking it safely inside the fatty lipid bubble of an exosome is an ideal way to transport it.
Got it.
And when they introduced those curcumin-loaded exosomes to the target cells and then turned on the ultrasound and the heat, the cellular machinery basically went into overdrive.
The mechanotransduction from the mechanical stress, combined with the thermal stress, specifically upregulated a protein pathway called RAB7B.
What does RAB7B do?
RAB7B acts as a master regulator for that clathrin-mediated endocytosis we discussed earlier. So the heat and sound didn't just melt the cell membrane, they actively signaled the cell's internal machinery to start aggressively building those clathrin cages and swallowing material from its environment.
And what the cell was aggressively swallowing was the curcumin poison pill. The study showed that this specific ultrasound heat combination drove the loaded exosomes straight into the deep interior of the target cells. It deposited the payload right next to the cell nuclei.
And the clinical outcomes of forcing that uptake are the ultimate proof of the concept here.
Yeah, the numbers are just staggering. The researchers observed approximately 40 times higher cancer cell death compared to just administering the drug without the ultrasound and heat preconditioning.
40 times.
40 times. And when they transitioned to animal models, this thermal ultrasound method suppressed tumor growth 10 times more effectively than the control groups.
That demonstrates the absolute power of combining targeted biology, the exosome vehicle, with targeted physics.
Yeah.
You are basically manipulating the environment to force the tumor to eagerly consume the exact weapon designed to destroy it.
So if we look at the physics, it feels like a complete system, right? We've used heat to break the masonry of the blood vessel wall. And we've used heat and physical force to melt the resistance of the cancer cell so it aggressively swallows the drug. But going through the final papers in the stack, there is a whole other dimension to this. Heat doesn't just change the physical state of the lipids. It changes the internal biology of the tumor.
It does. Heat acts as a systemic biological alarm.
And this is where the heat shock proteins come in.
Right. When any cell in your body is subjected to environmental stress, and specifically this 41 to 43 degree thermal stress, it panics. It recognizes that its environment is rapidly becoming hostile to its survival.
Okay.
So in response, the cell instantly ramps up production of a specific family of molecules known as heat shock proteins, or HSPs.
And the sources heavily focus on one specific protein called HSP70. What is HSP70 normally doing inside the cell?
Well, under normal conditions, HSP70 acts as a molecular chaperone.
A chaperone.
Yeah, when a cell gets hot, its internal proteins run the risk of denaturing or unfolding and losing their shape. HSP70 binds to these proteins and tries to physically hold them together to protect them from the heat.
That makes sense.
But here is where the biology shifts in our favor. The severe heat stress causes the tumor cells to dramatically accelerate the quantity of exosomes they secrete.
So they start ejecting these bubbles at a much higher rate.
Exactly. And they pack these newly secreted exosomes full of this HSP70 along with various chemokines. Chemokines are essentially chemical distress signals, or like biological breadcrumbs, that tell the body where a problem is occurring.
So the tumor cell is literally sweating out these panic-filled bubbles into the surrounding tissue.
Yes. And this is the critical twist. When HSP70 is inside the cell, it's a protector. But the moment it is ejected outside the cell, riding on the surface of an exosome, its function completely flips.
Wow.
To your body's immune system, extracellular HSP70 is a massive, glowing red danger signal.
So by heating the tumor, we accidentally force it to set off its own biological burglar alarm.
It is a profound vulnerability, and it introduces a truly revolutionary concept in oncology. These heat-stressed exosomes, coated in HSP70, essentially act as a highly potent, naturally generated tumor vaccine.
That concept of a self-generated vaccine is just fascinating to me. How does the immune system actually process that danger signal once it's released?
The first responders are the macrophages. You can think of macrophages as the immune system's heavy-duty garbage disposals. They patrol the tissue looking for debris and pathogens.
Right.
When these macrophages encounter the HSP70-loaded exosomes, they go into an absolute frenzy. The HSP70 binds to specific receptors on the surface of the macrophage, which massively enhances their phagocytosis, basically their physical ability to engulf and destroy foreign material.
So the heat essentially supercharges the garbage disposals.
It does, but the immune cascade goes much deeper than just the macrophages. The thermal stress, combined with the release of those chemokine breadcrumbs, heavily promotes the differentiation of naive CD8 T cells.
Naive T cells. Those are the immune system's special ops forces that hasn't been given a specific target yet, right?
That's a great way to think of them. They're circulating, waiting for specific assignment, and the HSP70 danger signal from the heated tumor provides that exact assignment.
Oh, wow.
It triggers those naive cells to transform into highly specialized cytotoxic killer T cells that are biologically programmed to hunt down and destroy those specific tumor cells.
So the heat isn't just a physical crowbar for delivering a nanomedicine. The heat forces the tumor to unmask its own identity, handing the immune system the exact molecular blueprint it needs to mount a targeted attack. It's an incredibly elegant, multi-pronged assault.
Which is exactly why researchers view mild hyperthermia not just as a delivery mechanism, but as a fundamental shift in how we might treat resilient tissues.
Okay, let's pull all of this together and summarize the incredible journey we've mapped out today.
Sounds good.
We started with the frustrating bottleneck of infiltration, right? How do we get a drug into a solid tumor? And the answer hidden in these sources is the application of a highly specific thermal dose, 41 to 43 degrees Celsius. First, this mild heat dismantles the trio and VE cadherin fortress of the blood vessels.
Yes.
It disrupts the master mason, collapses the actin scaffolding, and pries open massive 10-micrometer gaps, allowing our exosome delivery vehicles to flood deep into the tumor bed.
And once extravasation occurs, that same thermal energy physically melts the lipid armor of the target cancer cells. It lowers the activation energy barrier and synergizes with mechanical ultrasound forces to supercharge the RAB7B pathway.
Forcing the cancer cell to aggressively swallow those drug-loaded exosomes via clathrin-mediated endocytosis. And then, as the final devastating blow, this targeted heat triggers a deep biological panic within the tumor itself. It forces the cancer to eject exosomes coded in HSP70, a blazing danger signal that wakes up the macrophages and programs killer T cells to actively hunt the cancer down. It is basically an absolute masterclass in manipulating the physical environment to achieve a biological victory.
It really is a remarkable convergence of physics and biology. And looking at all these mechanisms together, it naturally pushes us to consider a much broader application.
Yeah, where does this lead us?
Well, right now, all the papers we reviewed are focused on oncology. We are using localized mild heat to aggressively pry open disorganized tumor vessels and force drug uptake. But think about the most notoriously strict, highly regulated barrier in the entire human body.
The blood-brain barrier.
Exactly. The endothelial cells in the brain are locked down vastly tighter than anywhere else, making it nearly impossible to deliver drugs to treat Alzheimer's, Parkinson's, or severe neurological conditions. But if focused ultrasound and mild heat can deliberately dismantle the trio and VE cadherin mortar in a tumor without permanently destroying the tissue, what if we apply this exact same thermal intervention to the brain?
Oh, wow.
Could we use a highly targeted, computer-guided beam of thermal ultrasound to temporarily and safely melt a small window in the blood-brain barrier? We could deliver neural regenerative exosomes or targeted therapies directly into specific coordinates of the brain and then simply turn the heat off, letting the barrier naturally rebuild itself 8 hours later.
That is incredible.
We could potentially treat the untreatable purely by mastering the application of local heat.
Using thermal physics to unlock the brain itself. That completely reshapes how we think about treating disease, moving far beyond just targeting cancer. Well, to everyone listening, thank you for joining us on this deep dive. Keep questioning the amazing hidden mechanics of the human body, and we will catch you next time.